Okay, in the previous video we talked about the viral delivery of the genes, and we said that there are some problems such as the triggering of the immune response of the body, integration which can lead to the development of the cancerous tumors, the problem with the time duration of this activity. For example, it works at the beginning, then it doesn't work, and the selectivity of the viruses, which is not that high, so they can go and bind to the other cells and infect them. And we also mentioned about the death of a person which died due to the massive trigger of the immune response of the body. And now we want to talk about the non-viral gene delivery. So instead of using the viruses to deliver the nucleic acids into the body, into the cells, we now are going to use another nanocarriers such as the liposomes or polymers, which are not as efficient as the viruses. They are less efficient because the viruses are synthesized by the nature, but the non-viral gene delivery, when we use such nanoparticles, have some advantages, such as the cost effectiveness. It's better. The availability of the liposomes and polymers is much more. Well, the induction of the immune system is less because we just told you that for the viruses the immune system is gonna be activated, and all these limitations which are overcome by the non-viral gene delivery make them to somehow be more effective, but not practically, let's say, be more let's say in the literature more researchers have been attracted towards the use of non-viral gene delivery, especially due to the death of the person that we mentioned in 1999, which made most of the researches about the viral gene delivery to be stopped. Okay, so let's talk about the material that we are going to encapsulate inside our nanocarriers to deliver inside the cells. Well, we were talking about delivering the nucleic acids. Well, plasmid and the siRNA, the siRNA, these are the two types of nucleic acids that we can encapsulate inside the nanocarriers to deliver into the nucleus. Plasmid is a DNA. This DNA can enter the nucleus and is capable to replicate, to modify the genetic code of the DNA which is already present into the nucleus. So this plasmid has to enter the nucleus. On the other hand, the siRNA, it just interferes with this procedure that I mentioned here. For the synthesis of the proteins, we mentioned that this process is gonna happen outside the nucleus. We just interfere with this messenger RNA to block the production of the polymers. So for the time that we encapsulate the siRNAs, there is no need to enter the nucleus of the cell. Well, you see here that if we want to deliver the nucleic acids inside the cells within an ex vivo technique, in which that if you remember, we said that we isolate the unhealthy cells through the ex vivo technique, and apply an electric field to make the cellular membrane more porous, or let's say more permeable, to deliver the nucleic acids inside the cell more easily. Well, these are the complexes that I mentioned about 2 or 3 times. You see here we have the plasmid DNAs and we have the siRNAs here. And we know that these are both negatively charged, so if we use a complexing material such as the polymers or liposomes, which are positively charged, finally we have the lipoplexes or the polyplexes. And what is the difference between these two? The only difference is the payload. Here we have the plasmid DNAs inside, and here we have the siRNAs inside. So these lipoplexes or polyplexes do not have to enter the nucleus because they have these siRNA payloads. We just mentioned that these are going to interfere with the messenger RNAs, which are responsible for expressing the certain type of protein. But on the other hand, if we encapsulate the plasmids inside the lipoplexes or polyplexes, these final structures, nanocarriers, have to enter the nucleus to modify the genetic code of the cells. Well, when we designed the nanocarriers, the first step for going towards the cell is the binding with the cellular membrane. Well, if you remember, we mentioned that the cellular membrane is negatively charged. So if the nanocarrier that we have designed has the net positive charge, it can interact electrostatically with this membrane, so the binding will be favored. But what does define the net charge of these nanocarriers? As we mentioned here, the payloads are negatively charged, but the complexing agents are positively charged. But the ratio between the payloads and the complexing agents will define the final charge of the material, of the nanocarrier. If the amount of charge of the payloads, negative charges, is going to be more than the positive charge of the complexing agent, so the final charge will be negative. So it is not going to help us for binding with the negative charge of the cellular membrane. So we have to make sure that the ratio between the complexing agent and the payload is in a way that the final nanocarrier is positively charged, so that it can interact better with the cellular membrane, which is negatively charged. On the other hand, if we don't want to play with the charge, we can play with the targeting ligands. For example, RGD, if you remember, we mentioned that is a type of material which helps the cells to bind towards each other, to feel the mechanical force between each other, so that they will survive. We mentioned these RGD, if you remember, in the section of the hydrogels, which were used for the tissue engineering. So these RGDs have affinity towards the cell membranes. What does it mean they have affinity? It means that they like the cellular membrane. So if we accommodate means if we equip, if we load the RGD on the surface of the nanoparticles, so they will finally have the RGD expressed on their outer surface, so it will help us to increase the affinity of these particles towards the cell membrane, so they will bind more easily towards the cell membrane and the endocytosis will be increased. So these are all about the endocytosis that we mentioned in the biobarrier lectures before. But you remember that we said the first step is the endocytosis, but because we want to deliver the payloads inside the nucleus, we have to skip the endosome. If you remember, after the endocytosis, there was a vesicle formed, and this vesicle contained all the materials which were internalized, which came down to the cell cytoplasm. So this endosome will try to, let's say, degrade the materials inside this by providing an acidic environment. So the pH of the endosome is gonna be decreased, so the number of H plus or protons inside this is gonna be increased. So we have to skip this step to make sure that the material that we have here is going to be safe, because finally they want to either go inside the nucleus or maybe they want to interfere with the mRNA, the messenger RNAs. So how can we skip the, let's say, endosome? Well, there is a process that we can use the polycationic complexing agents. These polycationic, they have the property that when they are in the endosome here, the endosome, in order to provide the acidic environment, will use these ATPase pumps to enter the H plus inside the endosome. But when the H plus is entered, they will be absorbed by the complexing material that we have, so they will be neutralized. On the other hand, another H plus will come inside the endosome to reach to the final acidic environment that we want to have here. But in order to maintain the neutrality of the endosome, another Cl- ions will enter, so the osmotic pressure inside the endosome is gonna be responsible for the, let's say, recall of the water inside the endosome. So at some points it will explode, it will rupture, and all the material will be released, and we have our nanocarrier, the lipoplex or the polyplex that we designed in this slide I showed you. so they will be free to go. But it is like a hypothesis, it is like a movie, it doesn't seem to be real. This is what was said by the professor. Well, if you read the explanation here, you will exactly understand what happens in this process. But we have also another mechanism, which is called the flip-flop mechanism, in which if we use a positively charged lipoplex, it can interact with the negatively charged endosome membrane, and both of them will fuse together, and within a so-called flip-flop mechanism, the nucleic acids inside the lipoplexes will be released and they are free to move. Here there is another mechanism for escaping the endosome. We have used the photosensitizers. These molecules are sensitive to the light. When we accommodate our materials on the surface of the endosomes, by illuminating the light onto these photosensitizers, they will undergo a conformational change, and the endosome layer will rupture, and as a matter of fact, the nucleic acids will be released. So this is the A. We accommodate on the endosome layer the photosensitizers, and after the endosome is down inside the cytoplasm, we illuminate them with the light, and it will rupture so that they will be released. Well, as we mentioned, for the plasmid which have to enter the nucleus, if you remember here, for the plasmid, I told you that they have to enter the nucleus to replicate and modify the genetic code, but the siRNAs, they have to perform their action, their responsibility just outside the nucleus. So when we are delivering the siRNAs, only escaping the endosome will be enough for us. But when the payload is not the siRNA, but is the plasmid RNAs, we have to enter the nucleus. But the problem is that the nucleus has the pores which only allow the molecules with the 50 kilodalton to enter the nucleus. And this is a problem, because the plasmids are larger than 50 kilodaltons. So this is one problem about entering the nucleus. In order to overcome this problem, some scientists have found that there are these receptors, named as the importins or transportins, which can be stimulated, and when they are stimulated, the pores will become larger and larger, and the nucleus membrane will become more permeable, so even the plasmid DNAs which we want to deliver inside the nucleus can enter. Okay, wait a second, I have to plug my charger. Okay, so now let's continue with the polymeric vectors. You see here that we have the polymers which are due to these NH2, they have the positive charge, so they can be used for complexing the nucleic acids, and by playing with different types of polymers we can make the hyperbranch polymers. For example, you see here in this picture that by the use of the cross-linking between the disulfides we have made the hyperbranch polymers for encapsulating the nucleic acids, and these disulfides can be cleaved by the some sort of enzymes within the endosome process. And you see here how the transfection efficiency, I told you transfection simply means delivering of the genes, has increased. This is also another example about using the, let's say, polymers for delivering the genes, and this is something important that we can spend some more minutes about it. You see here that we can polymerize the cyclodextrins to encapsulate the nucleic acids. Okay, you know that the nucleic acids are negatively charged, and by the positively charged cyclodextrins, as you see here, when we polymerize the cyclodextrins, they have the NH2 plus, so they are positively charged. They can encapsulate the nucleic acids. On the other hand, we can introduce the polyethylene glycol polymers which have the hydrophobic head on their terminals, and these hydrophobic heads can go inside the hydrophobic hole of these cyclodextrins. On the other hand, if we accommodate the other terminal of the polyethylene glycols with the targeting ligands, finally we have this molecule which enters the cyclodextrins. So not only we have the polymer PEG chains here which can help us for avoiding the opsonization and their recognition by the immune system of the body, we have also these targeting ligands on the end which will help us to increase the affinity of these cells. If you remember here, I told you that by using the targeting ligands such as RGD, we can increase the affinity of the cells towards the cellular membrane. So this is the final nanocarrier which is nicely designed. Now let's skip this, and you see here if we accommodate the polymers with the radio labels by measuring their activity, their, let's say, radioactivity, we can realize how much of these molecules have been internalized inside the nucleus. And this slide is about the ratio between the DNAs and the positive charged materials that I told you is important to define the final charge of the nanocarrier, and how we can tune the final charge of the nanocarrier. Okay, here we have the nanocarriers which have been recognized under the confocal microscopy to characterize the nucleic acids which have been stained by the fluorophores, because they can be recognized under the confocal microscopy, if you remember. Now let's go about let's go for the liposomes. Okay, if you remember, we mentioned that not only we can use the polymers for the production of the polyplexes, we can also use the liposomes for the production of the lipoplexes. Well, by changing the formation of the lipids, for example, changing the length of these hydrophobic tails or the hydrophilic heads, we can finally change the final shape of the self-assembly of the liposomes or the lipoplexes that we get. We can change the curve. For example, have a look at this picture. You see here that by tuning this, we can have these layers or these special formation of the lipoplexes to vary the curvature. So you see here another pictures of the AFM, pictures of the DNAs, the liposomes and the lipoplexes together. And the liposomes, in order to release the nucleic acids, they can do it. The release of the nucleic acids can be triggered even by the temperature, by getting to the target, or changing the pH, or applying an electric field. So these are different types for releasing the nucleic acids. And we can stop here because the other things that we have are not that important. Anyway, please download the notes that I have uploaded, and you will be able to follow the videos without taking the notes. And that's it. In the next video we will cover the lecture number 12.